Hi everybody, today you're going to learn about evolution and genetics of populations. So genetic drift is something that's caused by random change in allele frequencies happening by chance to small populations. So it's caused by non-selective pressures, so not natural selection, but just non-selective pressures like increased death rates or decreased birth rates, or natural disasters like a tornado or fire or volcano. And it's also caused by a bottleneck of a large population suddenly being reduced to a small population, like by hunting or something like that. So if you look at this picture right here, here you have a group of insects or ladybugs, and something happened to cause their population to go way down, and now the genetic variation of the new population is very different than the original. And then you can also see up here, these are two different populations. N equals 100, that's the number of individuals, and N equals 10. And then these are the frequencies of this B allele. So you can see that here it's pretty uniform and there's not a lot of variation. But here it goes all over the place because there's so few individuals, and that's what genetic drift does. The founder effect is also a random process that decreases genetic variation in a population. So that's when a small part of the population is removed from the larger population. And that can happen because of any kind of isolation, so migration or some kind of geologic event that separates a small population from a large population. So you can see that this was the original population of the ladybugs, and there's lots of variation, but then this is the new population, which is going to have less. So this is what happened when the finch went over to the Galapagos and started its own population. So migration and gene flow is where you have movement between two populations resulting in exchange of alleles. So like this one right here is going to this population, and it's going to exchange alleles and change the genetic makeup of the gene pool here. In short term, this introduces new genetic variation, right? Because this one is going to come over and give its genes, which are different from this original population. Over time, though, with gene flow between these two populations, the genetic variation between the populations is going to decrease, and they're going to become more similar because they're exchanging alleles. And you can think of that in the human population. Before travel was easy, right? The populations in different parts of the world were pretty isolated, and they were very different from each other. But now with travel being easy and technology, right, those populations are becoming more and more uniform and similar. So remember, populations evolve. It's not the species, it's a population, and it's the change in frequency of traits. And a gene pool is all of the genes in that population. So when a population is in genetic equilibrium, that means that the allele frequencies, like the big A's and little a's, are staying the same generation after generation, and the population is not changing. So that's what genetic equilibrium means. Now, there are 5 conditions for genetic equilibrium, and the first one is that there are no mutations. So we know that mutations happen all the time, and they're random. There's also random mating, which means no sexual selection. We also know that individuals almost always choose a mate because of different characteristics. The genes don't affect the survival or reproduction, which means there's no natural selection. There's only large populations, so there's no genetic drift, and there's no immigration or emigration out of the population, so there's no gene flow. So as you can tell, these conditions don't happen in nature, right? But they provide information so that we can make a null hypothesis. So all 5 of these have to be true for the population to be in genetic equilibrium. So Hardy-Weinberg is a way that we can calculate the genotype or phenotype frequencies, or the allele frequencies, if the population is in genetic equilibrium. So the equation that we use is right here, which you should write down. And the p squared represents the homozygous dominant frequency in the population. 2pq represents the heterozygous frequency in the population, and q squared represents the homozygous recessive frequency in the population. So if you're ever given a question and you need to find the genotype or phenotype frequencies, you're going to use this equation. You can also find the allele frequencies using this equation. So P plus Q equals 1, and P stands for the allele frequency of the big A, the dominant, and Q stands for the frequency of the little a. And remember, if there's only two alleles, they have to equal 100% of the alleles in that population. So make sure you write those two equations down, write what they represent, and then we'll do some practice problems. Okay, so here's our first practice problem. So evolution involves change in frequencies. For a particular gene locus, and that just means location, in a population, the frequency of the recessive allele is little a, is 0.4, and the frequency of the dominant allele is 0.6. So if you look down at this equation, right, P, which is 0.6, plus Q, 0.4, should equal 1, and it does. So the question is, what's the frequency of each genotype in this population, and what is the frequency of the dominant phenotype? So in order to find frequency of genotypes and phenotypes, we're going to use this equation. So we know that p squared is equal to big A. So we're going to take the p, which is 0.6, and we're going to square that. And then to find 2pq, which is your heterozygous frequency, we're going to take 2 times p, which is 0.6, times q, which is 0.4, and that's going to give us our heterozygous frequency. And then for our homozygous recessive, we're going to take q squared, which would be 0.4 squared. And all of those have to equal 1 because that's all of the genotypes in the population. So if you do the math, you should get this. Now, the dominant phenotype, remember, is going to include the homozygous dominant and the heterozygous. So to get the dominant phenotype, you just add these two together. If it said what is the frequency of the recessive phenotype, then it would just be equal to this one. All right, and that is it. We'll do some calculations in class. Make sure that you write down the equations and write down this example. Okay, thanks.